So good evening, ladies and gentlemen, and a warm welcome to yet another session of Expert Speaks. The world is going through tough times, challenging times, testing times currently, and India is not an exception to it. All that we can do today is, be patient and try to be safe and keep our near and dear ones safe. Apart from that, I really don't know how much is in our hands, but yes, this is something that we can do. We are all reading to several articles, several stories, and I would say that at some point of time there is an overdose or overload of information that is happening. We are googling out things, we are reading articles in the newspaper, on the media channels, and a lot of other stuff. And not to really miss on, we have our very famous WhatsApp university, the so-called university today. And we really don't know how authentic, how real, how true, how verified all this information is. We're really not aware of it. So I thought, why not take this opportunity and speak to someone who can give us the real, sort of the real story, the real things behind not just the pandemic or not just coronavirus, but somebody who will speak to us overall about the world of microbiology and what it all means and all of that. Very coincidentally, and I have to, before I start the session, I want to thank my very dear friend Arun Bedekar, who actually gave me a contact of Dr. Girish Mahajan, and when I'm going to read out his bio sketch, you will know who Dr. Girish Mahajan is. And I am really, really grateful to both these gentlemen, to Arun for connecting me to Dr. Mahajan, and to Dr. Mahajan for accepting my invitation to come on this show. Before we proceed, I would like to read Dr. Mahajan's bio sketch. And I'm saying read because the bio sketch is something, it is beyond my capacity to, to have by hearted it, because first of all, science and all these scientific terms and a vast experience and everything, you will hear it yourselves. So I will now start with the introduction of Dr. Girish Mahajan. So Dr. Girish Mahajan has a vast professional experience and was associated with various renowned organizations such as Piramal Enterprise, Omni Activists, Unilever, Hest, and Johnson & Johnson. He is an active member and mentor of the American Society of Microbiology and Association of Microbiologists in India. He is the member of Special Scientific and Technical Appraisal and Advisory Group on Marine Bioresources and Biotechnology Network by Government of India, Ministry of Science and Technology, Government of India, Fellow of Society of Applied Biotechnology, India, member of the Board of Studies and visiting faculty of various premier schools of biotechnology and microbiology. He is the alumni of ICT, Seawood Committee of India, and is also on the editorial and reviewer board of 6 international journals. He has 19 patents in his name, 19. He also has 137 international and national scientific publications, book chapters, media publications, posters. He's invited as lecturer. He has many national and international collaborations, joint venture coordination experience to his credit. This is not all, there is a lot more, and I am going to read all of it. I don't want to stop there. His core expertise has been drug discovery and development, especially anti-infective from microbial resources. There are certain things which I can't even pronounce, which I will leave for Dr. Mahajan to let us know about. But I still wanted to go further, not stop there, and still want to go further and say a lot more things about Dr. Mahajan. He has guided 27 postgraduate dissertation projects. He has contributed to many technical brainstorming meetings organized by CSIR, DBT, DST, etc. He has mentored several professionals and has been the counselor to many schools and colleges. He has been an excellent career guidance coach, especially in the area of microbiology, biotechnology, and biology field. Dr. Mahajan may not be an MBBS doctor, but what is he? He has acquired his doctorate in microbiology, and from where? From the famous Institute of Chemical Technology, formerly we used to call it UDCT. Institute of Chemical Technology, Mumbai, and developed a very cost-effective way of producing a high-value PUFA. Doctor, you will have to explain to us about this later, from
Sure.
marine algae. He has been honored with several prestigious national awards, such as the Distinguished Industrial Microbiologist Award 2020 by Microbiologist Society of India. He's also awarded AMI Louis Pasteur Award 2013, OPPI Scientist Award 2012, ISCB Award of Appreciation of Industry Scientist 2012 in the area of drug research. He's awarded the Eminent Biotechnologist 210 and ICT You've been visiting fellowship for bioprocesses technology 211 212 by ICT. He has been he's also been a scientific counselor for some schools and colleges for the biotechnology and microbiology profession, actively guiding budding scientists and he's been writing in the media. Currently, he's working at HiMedia laboratories, and Doctor will explain to us what he does there. Because again, there are some words which are very difficult for me to even pronounce. So it is Doctor who's going to speak to us about all of this in a short while from now. Without wasting much time, I would like to welcome Dr. Girish Mahajan on the show. Expert Speaks. Doctor, we couldn't have another expert like you here. You have tremendous experience to your under your cap. We will not waste much time and start with our question answer session, Doctor, if you're ready and comfortable.
Sure. Yeah.
Okay. So, Doctor, in your introduction, I kept using the word microbiology, microbiology, microbiology. So, my first question to you, what is microbiology? For a layperson, I'm a commerce graduate, by the way, and I'm sure a lot of my friends or family who's watching, they're also all not from the science background. So for lay people like us, laymen like us, how would you put what is microbiology?
Okay. First of all, Sonal, thank you very much for inviting me on this great forum of experts, which you are conducting for the social cause, and introducing the experts across the world. And also my special thanks to Arun Bedekar, the event manager of our Nirdhar group, and thanks to him to introducing to you. And so here am I, and so let me answer your first answer to your first question. Like of course, as I told you, like I'll be using some slides in between, because
Yes,
I'm talking about really invisible words, so
yes.
they are to be given some shape, so that the viewers and will be familiar with some how they actually look. So basically, if you ask like what is microbiology, and in a layman's language, so microbiology is basically, technically, if you ask me, it is a study of very tiny animals, microbes. They are called as. That's why I call microbes. The word micro has come because of the size of these microbes is varying from nanometer to micrometer. Now you may wonder what is nanometer to micrometer. Definitely, all of us have studied in the school time, but still, for the sake of viewers, I can say that micrometer, look, suppose if you have, all of us know 1 meter, right? So if this 1 meter is divided into 10 lakh parts, 10 lakh, okay,
Mhm.
that one part is called as 1 micrometer.
Okay.
And that 1 micrometer, if I divide further into 1000 small parts, it is called 1 nanometer. Now the size of these invisible entities varies from nanometer to micrometer.
Mhm.
Now again, though they are in nanometer to micrometer, the size and the shape and the beauty of them varies a lot. In fact, if you consider purely the microbiology, so I can classify, I mean, I will share one slide with you. Please allow me.
Yes, sure.
Can you see?
Yes, yes.
Yeah. So let me go for the size. I think since all of you are viewing very closely, you can easily see the unit here. It starts with 0.1 nanometer, which is the size of the atoms. 1 nanometer amino acid molecules. Then 10 nanometer up to 100. 100 nanometer is the field where our today's famous coronavirus 80 to 120 nanometer. This is the scale. If you go further, 1 micrometer to 10 micrometer, all our bacteria, useful, harmful, are sitting here. On the same scale, if you go 100 micrometer, we have certain higher protozoa like amoeba. They lie in this belt. Then further, there are large protozoa, human eggs. Then 10 millimeter is a tick bug, and 100 which is 10 centimeter, our heart size. 1 meter is a dog, like this. So this scale will explain you how tiny are the microbes. But instead of they are tiny, they have a beautiful shapes. You can see some like pictures I have collected for the all of us. Like these are the shape of the bacteria. Some bacteria are cocci. These are called cocci. They look like bundi. And this is a rod-shaped bacillus. It is called. And the spiral one are called as spiral-shaped spirulina, or it could be trichonema. Then there are viruses. Like I can say microbiology as such, as I told you, the word microbes means small animals. That's why the study of these microbes is called as a microbiology. And as such, microbes include basically bacteria, fungi, viruses, protozoa, and algae. So some representative I had shown here. So these are the viruses. Again, in spite of they are in nanometer size, they are different size and shapes. Nowadays, this shape is very famous because of this coronavirus, RNA virus.
Yes.
But there are phages which look like as if satellite. And to just tell you something more, this phage are so small that this one phage can attack a bacteria. You can see the bacteria, a green color. It can attack this bacteria, and one bacteria can accommodate 3,000 to 10,000 such phages. So from that, you can imagine how small the viruses are. On this side, you can see certain fungi, penicillium, aspergillus. I think most of us are familiar about yeast.
Yes.
And mushrooms are again very famous. And of course, mushrooms, though they are called microbes, mushrooms are a special category which are very much visible to naked eyes, and they can be cultivated and found in the forestries, etc. And as such, microbiology is a long history. It's about 345 years old history. It started with the actually Anthony Leeuwenhoek. You must have heard the name sometime in the schools. Like the whole story, microbes, people were using microbes, but unknowingly. But when this Anthony Leeuwenhoek, this fellow, he discovered with a serendipity, means accidental discovery, I can say, in 1676, the story of microbiology began. So I think that's all about I can say at present about this microbiology. So perhaps we can switch over to next question. Or if you have any other question, sub question, you can ask me.
Yeah, so a little more in detail about microbes. You just told us about it, but maybe a little more in detail about microbes. And so viruses are a part of these microbes, or
Yes,
so
viruses are
virus,
the part of these
bacteria,
microbes.
all of these are parts of these microbes.
Yes, viruses are the part of the microbes which are very small in size. Now, how these viruses are different from other microbes? Okay. Now, viruses are actually when they were discovered, like that in the scientific community, there was a great dilemma that whether they are living organism or dead organism. So they are called as a borderline of living and dead organism, because we can crystallize certain viruses like tobacco mosaic virus, TMV. Tobacco mosaic virus is a virus which attacks the tobacco plants. Tobacco plants, they have a very long leaves, and they cause a browning of those leaves. And these can be actually crystallized and kept for years in a something like called Barani, glass vessels. And again, if you infect the leaves, it will reinfect.
Okay.
So people are wondering, and what are the typical life when you say particular organisms are living? It has to reproduce. It should have its own metabolism. It should have a cellular structure, like there should be cell wall, nucleus, etc. But viruses, they do not have something called cell wall. They do not have cytoplasm. These are the most common terms, which we often hear from our school days. But viruses lack such cytoplasm. Viruses lack such nucleus. There is no such thing called, division, etc. Then how do they survive? That's a wonderful. What do they do? So first of all, most of the viruses, they are the pathogenic parasites. Means they need another host for the survival, because they cannot complete their own living machinery on their own. Okay, now take example of say coronavirus, which all of us are very familiar and well educated through WhatsApp and media. Like this creature of 80 to 120 nanometer size, now you can understand nanometer how small it is. So when it attacks through certain receptor, this virus has spikes. Now what is a spike? It has a thorn like thing, So with that thorn on its surface, it can go to the guard, we can say receptor on our cell, on the lung cell or other cell. It asks them to open the door. And when this receptor opens the door, it pushes its RNA inside our nucleus, inside our cell. And this RNA, now what is RNA? Now RNA or DNA, till now all of us know DNA as our life code. But in this case, it is a little bit different. RNA is their life code. So you must have heard DNA fingerprint, DNA forensic medicine. But in case of virus, RNA is the life code. And this RNA is pushed inside the cell. And then it catches hold of the machinery of the cell, our cell. Now see how the beautiful it is. It catch hold of the machinery of our cells, and it makes us to cook the recipe for itself. Now it needs its body parts, it needs proteins. All these are synthesized using our cell nucleus. It means that's why it is called as a parasite. And suddenly our cell will produce large number of particles of this SARS-CoV-2 coronavirus. And same is true for everything. So all virus mode of action is similar, but the doors, the receptors are different, and the way they attacks are different. In this case, it is called spike protein. So this is the way like viruses attack, and once there, you can say like baby viruses are produced inside. So obviously it will come out, the cell bursts and come out. But during this, our body invites lot of WBCs and other platelets. So the whole cascade of inflammation get activated, so that they want to kill the virus. So this cascade of anti-inflammatory things which is activated in our body, that causes a lot of symptoms. That coughness, fever is a reaction given by the body to the viruses. So this is the way viruses attack. Now suppose if I take this virus and put in a glass bottle, it may remain for years. Of course, for this there is no data, but I'm saying example of the other viruses. So they can remain for years, and again if I take out from the bottle and infect a particular tissue, they will start the infection. Now other microbes like bacteria, fungi, they have their own cells, they have their own DNA, they have their own ability to reproduce. So they want just some organic matter, like on bread the mold grows, fungus grows. And if on the roots or in any other like milk, many bacteria grows if you keep it open for some time without boiling or heating, so it gets spoiled, because many bacteria which are present outside, they grow easily because they have their own life cycle and own machinery to work on it. So this is how the viruses are different. And again, as I told you, size is very tiny. In a single bacteria, 3000 viruses can reside. So from that, you can imagine how tiny these viruses are. So of course, and they have the gadgets, but these gadgets are just to attack the host and complete their life cycle, nothing else. And of course, in other microbes like bacteria, the gadgets are a little bit different. They have the flagella, they have the pili, and they have sporangium. They produce spores. Nothing. Viruses are very silent killers. So they have just certain spike proteins, attack, kill it. But they cause a disaster. But on the positive side, I would also say, not to get flowed more into corona, like overall in the microbiology it is said that only 1% microbes are harmful, but rest other microbe there is a tremendous use and a huge market. I will show you some, share you some data soon, like how these microbes are useful, and without that we really cannot survive. So, that's how I can explain.
So there's one question here.
Yeah,
So the question is, so it is said that viruses are non-living unless it enters a human body. So what makes a
right.
virus use this mechanism? Why does it always require a host system to multiply and proliferate? This is a question asked by Shriya Fadnis.
Okay, Shreya, it's a very nice and technical question actually. And see, all this in case of viruses, it is nothing but a packet of chemicals. You can say packet of bad chemical. So whenever it attacks something, there is a complementary, like suppose key and lock mechanism. If there is a key, for every key there is a complementary lock, or I will say for every lock there is a complementary key. In the same way, whenever a virus, it has a spike, it gets complementary receptor. Like in case of corona, they call ACE receptor. If that receptor, they bind together, they have a locking mechanism. They lock, and the reaction cascade is initiated. Like in some of those, like if you keep the playing cards, like in this position continuously, and if you tickle one playing card, all the cards will fall. Tuck, 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 tuck.
The
Now this is called
dominoes
cascade.
effect,
Yeah.
something
So
like the
exactly
dominoes
same
effect.
way. Correct. So exactly same way, this cascade takes place, and it is not that why does it do? It is a complementary chemical which is available. If that complementary receptor is not there, it will not attack. Simple. Like in small kids, virus don't attack. This corona especially, because the ACE receptors are very sparse, or there are no very sparse things are there. It all depends on like receptors which are present on them. And one more thing, what you asked is why does it require a host? Yeah,
Yeah,
because
require.
the genome size, the genetic code size of this virus or any virus is so small that it cannot accommodate everything. Like in home, if you have something to make, say, pasta, but you don't have, say, mayonnaise, you don't have certain spice. Now, obviously, what you're going to do, you're going to buy some house or shop and go back there and get the things. Exactly in the same way, this virus has certain RNA, certain codes, genes responsible for production of its protein, production of its nucleus, but it does not have the mechanism to produce those enzymes. It does not have a promoter gene to produce those enzymes. So this is taken by this particular host, and host has like our body cells are complete package, so that they can easily provide everything. So virus nicely goes into our kitchen, our nucleus, and uses the mechanism and cooks its own capsids and bursts the cell. So it is like ungrateful entity. It kills our cell. I hope that answers your question, Shreya.
Shreya, I hope your question is answered. Uh, So, we're speaking about you told us about the size of the microbes and the size of these viruses and all that we heard. Doctor, now they're not visible to the naked eyes. How
Right.
to prove their existence then? How do you prove their existence and moreover identify the way we identify plants and animals? How do you know which organism is what sort of a virus or because there's so much of buzz today also around this coronavirus, false positive and false negative, and all those things. So similarly, how can we prove their existence?
Okay. Since you asked about how you identify the microbes, so it's a little bit broader question. I would
Mhm.
like to share both the parts, coronavirus as well as other parts. Now, if you say, if all of us are sitting in front of mobile and laptop, currently, if you look at your hands, you will feel, oh, it's very clean, nice. But if you take the thumb impression, single thumb impression of this on a very rich media. Now, when I'm talking media, it is not our social media. It is a media means it is a nutrient media, which is required for the growth of the microbes. Now, if I bring a plate in front of you and take a thumb impression, the microbes will start growing fast, like bacteria. So bacteria, single bacteria, you can say within 7 hours, it becomes 20 million in the numbers. So obviously, all these 20 millions cannot spread out. They form a so-called colony of the microbes. So at this moment, I would like to share one slide so that you will understand it better, what I'm trying to say. So, can you see this whitish thing, colony?
Yes, yes.
So, this colony, now a single, this is a colony of bacteria, and on the right side, you can see the colony of the fungi.
Mhm.
Now, a single colony can have 10 raised to 12 to 10 raised to 14 bacterial. So, we can see now these colonies are visible. But if you ask me a single entity, it is not visible. Now, you asked, how do you identify them? So, if I go back to the same side, I can say for bacteria and fungi, we use different nutrient media, which we call it as a meals of the microbes, because it's a living entity. It needs certain feed to grow. So obviously, we have to provide the feed in a liquid form, semi-solid form. Now, these could be from the synthetic or natural origin or a very, functional types. Like I would definitely like to say, the recipes of microbes could be so varied. You can see some basic ordinary media. It is like typical thali. If you go to hotel, here I spread jalamanta taprevza. Then we have enriched media. Okay, certain bacteria they can't live without blood components. They need certain blood component in that. There are enrichment media. Like some people say, after meals I want coffee, tea. So based on that, it goes ahead. Selective media. Now certain people say no, यार, I'll prefer non-veg only. मला तर chicken मटन जमतच नाही, दुसरं काही हातच नाही मी. Selective. Okay, you get this. If I incorporate this, obviously the selective will grow, non-selective will disappear. Like on a table, if you keep chicken mutton, so non-veg will jump on it, veg will disappear. Same way happens here. Differential media, like in certain nutrient media, we put indicator, and based on the type of microbe goes, we see different colors. And there are certain media which are selective as well as differential media. There are certain microbes which do not require oxygen for their growth. That is one of the miracle. Like these microbes can survive and produce colonies without oxygen. And one of the best example, which all of you are aware unknowingly that you are having that anaerobic bacteria, is a tetanus. Whenever some accident takes place, even if you fall somewhere, come in contact with the soil, your mother, father, or parents often say, tetanus injection घेतलंस का? So
Correct.
that tetanus is a bacteria, and it is a hardcore anaerobe, but it can cause horrible things in our body. And there are certain transport media, like in case of corona, you must have heard viral transport media, VTM. When your sample is collected from the nasal respiratory tract, through the swabs, it is kept into some red color solution. That red color solution is called as a transport media. So what I'm trying to say is that there are so many ways by which we can identify bacteria, fungi. Now coming to the entities like corona and rotavirus group or HIV, so these are difficult to grow on any such organic media. They cannot use. They are hardcore dished, you can say, animals, like carnivorous animals. They need living tissue for cultivation of them. So they are cultivated on a specific cell lines. Means you must have heard in Marathi called Naramasa Bhakshak Vag. you know.
Oh.
So Naramasa Bhakshak Vag Naramasas lagta. Without that, it cannot grow. In the same way, particular virus needs a particular tissue. So if I cultivate the tissue on the plate, on that, if you put that, it will nicely get cultivated and we grow, and you get a good number of the particles. So this is how they are cultivated. Now the next stage comes, because to identify we have to cultivate them first. Now identification, there are many mechanisms, but to simplify I can say every bacteria, fungi and viruses, they have a conserved sequence of the DNA, and in case of conserved sequence of the RNA. So there are certain techniques, molecular techniques, by which we take out their gene and look for certain probes like 16S RNA, 18S RNA, and then we decide, okay, this is say E. coli, this is Bacillus, this is Mycobacterium tuberculosis, or this is rabies virus, or this is SARS virus. So like this, so identification is today based on the molecular mechanism. But prior to that, we have to cultivate them and having a number of amount, and then you can do this next stage. Like one example is RT-PCR, which is being done from the patient. What do they collect? They collect the sample, and what the sample has, it has the live viruses particles. Now these samples are carried to the lab, and what they do is they rupture the virus. They take out the RNA, and there is a polymerase, reverse transcriptase polymerase gene. PCR means polymerase chain reaction. So in this polymerase chain reaction, they produce a multiple copies of their genome. First of all, that RNA is transcribed to DNA, and then that DNA checked for a particular genome which is specific for SARS-CoV-2, and through RT-PCR they produce multiple copies, and these copies of DNA which are generated, they are checked through the probe system. Okay, this probe is for SARS-CoV-2, this probe is for only SARS, which was seen in 2012 in China, and one is for MERS corona. No, there are 7 types of coronas.
MERS
So this is
was the one which had come in the Middle East countries?
Yeah, Middle East countries. Yes, yeah, that has, I mean, very high mortality, but the infectivity contagiosity was not so high. That's why it doesn't spread much. Means they were of the same family. So that's how today the identification part is done by the molecular. So everything is invisible. Nothing can be seen. Okay, you cannot take in hand. Okay, this is how it looks. No, everything has to be interpreted very intellectually.
Yes.
And sometimes people, sometimes they spread rumor. Arey, coronavirus ayes nahi hai. They see a lot of videos and this there is no coronavirus. Then how these things are happening? We can see in the electron microscopy the images. So I mean, that's a different case I'm telling, but this is how actually it is done.
Correct. I think Mr. Santosh Naik, your question is answered. Your question was, what are different medias of spreading viruses?
Sorry?
He had a question, what are different medias of spreading viruses?
See, you are talking only about coronavirus, then currently it is...
He's speaking about various viruses, of spreading viruses.
Yeah. See, viruses as such can be spread through many media. Like, first of all, many viruses go through the contact. It could be direct in coming in touch with the patient's contact. It could be air, it could be water, and it could be sexually transmitted. Like HIV is a sexually transmitted, or there are certain, in case of HIV only, I can say blood components. Whenever there's a blood transfusion, again it gets into the body. It could be sometime to the pet, like rabies virus. So there are different ways through which viruses can spread among the human.
So basically, we say, all these things we hear that bacteria or viruses, they are our enemies. Most of the time, we are always very scared of these bacteria and enemies and all, and because we hear that, kai infections hala ki bacterial infection hai, viral infection hai. That
Right,
is what we always keep hearing. So we, as a layman, again, I would always consider, bacteria and virus to be an enemy.
yeah,
But I also know that certain bacteria are required to make substances like आपला दही बनवताना किंवा वगैरे असं
barabar.
those are the times when you require. So can you tell us about the applications of these viruses and bacteria and how they actually work, or how they can be useful or how they
Right.
can be harmful both?
Yeah, definitely. I think since there is a lot of negative about viruses and microbes, I would like to also present you the very strong and which can really build nation's economy. That component also, like applications of microbes. So, but allow me to share some slides so that
Yes.
I can reach you all very closely with these slides. Okay. First of all, I will not say application, but a
Mhm.
simple like mechanism. Now, bacteria are the only entities in the world, on the globe, at least on the earth, which can fix atmospheric nitrogen. Now, you may wonder, what do you mean by this? So, in the air, there is 80% nitrogen and about 18 to 19% oxygen. Now, our body requires nitrogen because our body contains muscles, bones. All this contains nitrogenous compounds called amino acids, or rather, I can say proteins,
Mhm.
vitamins. All these contain nitrogen. The thing is, if I ask you from where the nitrogen comes, so definitely all of you will answer me, it's coming from food,
Food.
whatever, veg, non-veg you are eating. Now, next question is, from where the nitrogen comes in that food? Obviously, the food is either animal or plant. Let's say example of plant. The nitrogen is coming from the plant. Next question is, from the in the plant where the nitrogen comes. So one can give easy answer. उसमें क्या है? Soil. Now the next question is, from the in the soil, from where the nitrogen comes.
Correct.
Now in the soil, nitrogen comes only because of bacteria.
Okay.
There are species like Azotobacter, Rhizobium. No, so these bacteria and certain blue green algae, which have a tremendous ability to convert atmospheric nitrogen into very good nitrogenous compounds in the form of nitrogen fertilizer, and these fertilizers which are available in the soil is consumed by the plants. Plants are consumed by animals, both animals and plants are consumed by a human,
Human being.
and then we get our proteins. Now, has been there is a situation like, if think of a day there are no microbes in the world, nothing can be fixed from the atmosphere. We have to generate huge mechanism to fix atmospheric nitrogen. There is, because there is no natural way other than microbes. So that is the first natural way because of which we can survive peacefully on the earth, because these microbes are bringing that. So it's a big boon that they are doing the work of nitrogen fixation unknowingly for us. Second is biodegradation, energy production. Now, lot of sewage we generate, in cities, in villages. Now, the wonder thing is, daily we put our kachra outside the home, and from the society, tons of, this waste is collected. Where does it go? All this sewage and this waste is actually degraded by the microbes. Degrade means decompose. कुजण्याची प्रक्रिया होते. So this degradation is possible only because microbe. Because there is no other one who will going to degrade. There is no human entity or there is no human agency that they can bring about degradation by themselves. No. Whenever there is an oil slick in the oceans through the leakages, these oils in the ocean are again degraded. Who does that? Microbes. So they have a tremendous application. Energy production, again, microbes, like energy in this form of alcohol production that can be done with the help of microbes. One big business, like I am trying to tell you like how microbes are beneficial. Amino acids is one of the biggest compound, which is a component of a protein, glutamic acid, lysine. So these amino acids are produced by the microbes, and they are produced in many lakhs liters of huge fermenters. So we call it as a Kinman Prakriya. So in these fermenters, these microbes are cultivated, and amino acids like glutamate, lysine are purified and supplied to the industry and various food industries. Now, one more very familiar name, and you can say today, whatever RT-PCR we are doing and saving the millions and testing, it is possible because of one microbe called, Thermus aquaticus. You may wonder why. This Thermus aquaticus is a thermophilic bacteria. Now, this is a bacteria which can survive temperature above 100 degrees Celsius. Means boiling water also it can easily survive. It can come out, it can grow nicely. Now, when scientists studied this particular bacteria, Thermus aquaticus, they found that it has a particular enzyme which is called Taq polymerase. And this Taq polymerase is a key driver in PCR reaction. Now, it's a big industry today. The millionaire become billionaire who is producing Taq polymerase in this era. Now, this Taq polymerase is an enzyme produced by this bacteria which can tolerate a temperature of 95 to 100 degrees Celsius. And there is a step in PCR where the temperature requirement is above 90 degree. Now, if we would not have this particular enzyme, either we had to rework on PCR so that it will operate at low temperature, but it was not possible. So Taq polymerase has played a tremendous business in this. And of course, there are other enzymes, DNA ligase, restriction enzymes, which are useful in complete genome sequencing, creating artificial genomes of the bacteria, human. We can see the amount of business they are giving us. 12.7 billion 2019. 2020 चं पाहिलं असतं तर ते डबल टिबल झालं असतं. Now, citric acid. Citric acid is a very small component. It is also present in our regular eatable like farsan. Farsan खाल्ल्यानंतर शेवटी एकशे पांढरे पांढरे दाणे राहतात बघा. So if you consume that, you will feel it is the crystals of the lemon. But these are the citric acids. Citric acid is produced, a particular isomer of that, by a fungus called as Aspergillus niger. Now, one of the biggest market in which microbes are helped are all our antibiotics. Take the name of any mycin, kanamycin, annamycin. All these mycins, along with penicillin, ampicillin, and gentamicin, are produced by nothing else but microbes. And as for the prediction, the market is $45.31 billion, huge. Now, when I say antimicrobial, it includes both antibacterial, antiviral, antiparasitic agent, antifungal. We easily consume the tablets given by the doctor and get well. But behind that tablet, there is a microbe which is there to produce. And today we are semi-synthesizing the antibiotics. Like partly it is produced by microbes, and further by chemical reaction, we bring about some changes and make it more palatable. It's a list of antibiotics, famous antibiotics, of course.
Yeah.
And one more very interesting part, which all of you, or especially if there are some students in this crowd, it could be enchanting, that microbes can produce perfume, and this is a very lucrative field. And just to name a few, almond type of perfume, which is due to benzaldehyde, a specific enantiomer of benzaldehyde, it is due to a microbe produced by a microbe, and it is also called as a bracket fungus growing on the trees. Apple and pineapple like perfume, it is due to the butyric acid, and it is produced by Clostridium bitter. There is a list of such beautiful perfumes produced by, and in France, luckily there is a university and there are companies who are working on this. So perhaps in India, some of you can get, you know, starter of this type of thing. And of course,
Yeah,
there are research groups who are working on this.
correct.
One more area I can showcase you about the colors, because today everybody needs natural color, because these chemical colors, they have a carcinogenic properties, mutagenic property, genotoxicity. So that's why people prefer more natural product. And microbes are the best examples which are producing micro. We can see they are producing all 7 spectra of the colors. Like some example, like if you take any, you can say multivitamin capsule for eyes, you will see this name astaxanthin. There are certain blue-green bacteria which are producing phycocyanin, which is sold as Lima blue. It is used in the cosmetic for the blushers, tender blushers. So there are many examples like microbes are producing these pigments and people are using them. And recently there are certain pigments which are found antiviral, antibacterial. They are being used to color the masks also. Means people have approached us. One more very yummy part of these microbes is there are certain bacteria which are producing polysaccharide. I think all of you enjoy this confectionaries, chocolates, Cadbury's, and yogurts, and wonder why this yogurt is so yummy and so uniform, so sticky also sometimes. All the jelly-like properties are because of certain polysaccharides, which are called as a hydrocolloids. So this alginate, cardlan, dextran, gelangum, all these are used in various confectionery industries, food industries. And yogurt, when you bring from outside, you can check the composition. Somewhere you will find gelangum. Same way, if you eat any confectionaries, you check the label, there is a mention of dextran or cardlan. So all these are produced by this bacteria. And how they are cultivating them, they need the lakhs of liters of fermenters are available for cultivation of these microbes. So these are some areas: alginate, dextran, gelan, xanthan, confectionaries, beverages, soups. Sometimes when we make a soup at home, it is not as smooth as found in XYZ hotel. So this is because they add certain polysaccharide like alginate. It make it very smooth. So you can say all this is possible with microbes. One more very interesting field means if there are certain builders in this crowd, they may get attracted, or it's a topic of interest for everybody. Like many a times we can see in our home, outside our building, cracks are produced in the concrete.
Yes.
And these concretes are, I mean, it's a big loss to the society. They have to renovate, put the bamboos, and again restructure everything, repaint it. Now, instead of that, last 10 years, there are certain groups who are working on it. These cracks are filled with urea, and there are specific bacteria which are producing calcium carbonate deposition. Now, when a person, our contractor, fills these gaps, he goes at a 0.mm level perhaps or mm level. But these microbes grow at a micro level. So if you allow these microbes to grow, they will precipitate calcium carbonate deep inside these cracks and they will heal. And there is again research going on in this area, and even to our company, some people are approached like, can we make it faster? So of course, because of Corona, the things have a little bit lagged behind. So it's a big business now and going to be the future. One more big area where microbes are useful is the plant probiotics. Now, probiotics, all of us take many a times. We enjoy the probiotic juices, probiotic capsule, and most of the time, doctors provide whenever they give you amoxicillin or the penicillin derivative, they provide you one capsule. On this, it is probiotics. Okay, so it is nothing but a type of bacteria. Similarly, plants are also required to feed with microbes, which you call it as a probiotics. Like what did they do? They synthesize nitrogenous compounds from the atmospheric nitrogen. They solubilize the phosphate, they solubilize the potassium, and they chelate the iron, and all these are made available for the plant roots. So sometimes we call certain soils as useless. It's a dead, dormant soil. त्याला आपण
Yes.
नापीक म्हणतो. So such soil, they become dead or dormant because of the non-soluble things in it. But if you provide optimum content of microbes and moisture, they can be again made fertile.
Mm-hmm.
So from napik to supik. And of course, there are again huge areas. For the interest, I can restrict to some of these, like wine, beer, alcohol are very age-old. Vinegar
Yes.
manufacture, edible fungi like mushroom. I can give an example of mushroom. Recently
Yes.
in the newspaper, like Pradnesh Patel, a graduate who become a farmer, and he started cultivating microbe. These are the actual photos from his
Mm-hmm.
factory, and he's earning 4.2 lakh per year. And
Mm-hmm.
now, as a graduate, you can understand to earn this much is not possible
Absolutely.
in today's
Absolutely.
era. So it is a big business. And spirulina. So you asked about that
Mhm.
word spirulina, which I had told you, PUFA, PUFA. So
Yeah.
PUFA means
PUSA.
polyunsaturated. Yeah, that PUFA is polyunsaturated fatty acid.
Okay.
This spirulina is actually you can call it as a magical algae, microalgae, which
Okay.
is cultivated. So on which I had done actually PhD. So
Oh.
I proved mass cultivation for the PUFA. It is called as a gamma 6 type of fatty acid, omega 6 type of fatty acid. So
Mhm.
this is again a business like by which we can produce spirulina powder, and a farmer
Mhm.
can earn up to 40 to 45,000 per month. So I think this plethora of this applications of microbes, I mean, definitely tell you how useful the microbes are.
Yes, are
And of
useful,
course,
not just our enemies.
yeah, yeah,
Yes. Otherwise बॅक्टेरिया आणि व्हायरस म्हटलं की एवढी भीती वाटते की ते
exactly.
but yes, there as you have shown us and as we have learned. Sir, from this all these slides when you were showing, the thought that kept coming to my mind over and over again is that everything that we say is organic, organic food, organic, perfumes, organic material. I think for all of that, somewhere or the other, these microbes are instrumental
Yeah,
in making it to the real organic substance, rather than
perfect.
using some
Yes,
different chemicals and making them, and we don't know the side effects. The best way is to use these microbes and develop, cultivate all this in the organic manner, is something that I
yeah,
felt,
yeah.
it's the message that I got typically. So, doctor, we are speaking and we are hearing and we are literally living in this era of pandemic. It's been a year, more than a year now, and we're still here. So, and coronavirus is something that we've been talking about, hearing about. What exactly is this coronavirus? So, before we talk about this, just a little bit. My son is sitting next to me. He may say, "What is this that you're talking about?" But when this COVID thing came and all, he used to tell us that he's an MBBS doctor, so he said, आमच्या MBBS च्या पुस्तकामध्ये तो एक छोटा paragraph होता COVID वरचा, which we never even bothered to read about it, and now suddenly this entire virus has taken the entire world with a big storm. And so what exactly is this coronavirus? परत त्याचे काही म्हणे वेगळे strains असतात, mutants असतात. So what are these mutants? How do they develop? Can you just give us a little bit of information on all of this?
Okay, sure. So, as I told you before, like coronavirus is a family of virus. In fact, there
Mhm.
are 6 types. In certain reference, they say 7 types of coronaviruses. And one property of this, now this particular coronavirus is of the size 80 to 120 nanometer, and it has a typical spherical shape. And this virus, it has basically 2 components. One is a capsid, I mean, there's a surface versus spherical sato, and on it, it has a spikes of the glycoproteins. So, which are the glyco means sugar, proteins means it is a polymer of the amino acids. So हे दोघांचं मिश्रण झालं, it is called as a glycoprotein. The spikes or the thorns are made up of these, but these thorns are very specific. These are the keys by which they can enter the receptor, or they can open the receptor and enter into the host cell. Now, in this uniqueness of this particular coronavirus is that it also has RNA as its nucleus, or as its, you can say, coding component. Means living things are, they are considered as all living things have DNA. But no, that diagma is totally different. So living things have RNA also as their nuclear core. Now, as the science has progressed, certain things have changed. Like once upon a time, like people have never thought about virus, but there are certain subparticles which are smaller than viruses also today. And like example, virus, which is a very slow virus, but coronavirus compared to that, it is a, you can say, a bit bigger in size. Now, coronavirus, it needs a particular host, the lung cell, you can put in a simple way, for its growth and for the cultivation. And one of the special property of this particular coronavirus is, or any virus we can say, whenever they enter from one host to another host, some changes takes place into their sequences.
Okay.
And these sequences change in the RNA sequence or the reverse DNA sequence is called as a mutation. And usually these mutations are harmless because they are not targeted mutations. They happen spontaneously. So these mutations sometimes work very harmful. Like if they occur in certain sensitive area, like in this case today, I think all of us are hearing about double mutation, correct? So double mutation means there are two different mutants of viruses which are entering into the same host, human, and they are giving out to a new third mutant. Like in Maharashtra, like we are hearing about two mutants, like one is called E484Q and second is called as L45-2R. Now these two mutants, the first one is from Brazil, South Africa, UK strain, and second is from California, US strain. Now these two, they are infecting and they are combining to give one more mutant, which is called as a B1617. Now this B161.617 is particular mutant is called as a double mutant because it is formed because the combination of the two mutants. Now this mutant has certain changes in its spike, but of course still we are not aware whether it can be really taken care by the current ongoing vaccines. There are good speculations, but still question remains. So any virus, even coronavirus, has ability to form its own mutations. In fact, I can say that all RNA viruses have a very stupid habit of undergoing mutation. That's why it is very difficult to have a unique vaccine for a particular RNA type of viruses. Of course, there are some scientific reasons for it. For DNA viruses, vaccine is a bit easy. The mutation frequency is also very less because they are under DNA viruses are strictly controlled under DNA related enzymes. Of course, there is an intrinsic mechanism which is beyond the scope to explain here, but I can say RNA viruses have this ability to undergo quick mutation because of the less control during their multiplication. So that's why it is going to bound. Like one example, I can use common cold virus, which is of the again a RNA virus only. Now there is no vaccine till now for common cold. सर्दी झाली की 2-3 दिवस आपल्याला काढावे लागतातच. Yeah, same way this is also same category virus. And as I told you, it has originated from Wuhan as per the current knowledge through the wet market, and then it has spread very fast, and contagiosity is its the highest way by which it can spread. You can say based on current data, this is one of the most contagious entity found on the globe up till now.
How does it affect the body? Because death rate also we hear that it's on the high. So what exactly does it do to the body that people are dying of it?
See, the death again, there are two mechanisms. I mean, the two ways. One is it is not 100% the virus always responsible. The death is also because of the immunity power of the person. Like it is not that everybody who is getting affected go to severity. It
Yes.
ranges from asymptomatic to the death.
Hmm.
Okay. Now the death cause is mainly what is discussed in the medical science is that it actually activates the anaphylactic or allergic cascade system so severely and creates a cytokine storm in the body. And all these cytokines and anti-inflammatory things, they are produced so vigorously that it is like an autoallergic system created inside the body. So this gives signals to the organs and lungs and especially heart. So they get badly affected. And again, the death cause means can be explained in the medical way. Not you can say up to virus. I can tell you about this part, but anaphylactic and allergic component, they are the main
Mhm.
which are affecting this, and they are bringing about the, you can say, bad phase of the human body.
Mhm.
But within that, if you get controlled, then it's good. Like simple, you can say getting fever. It is again a mechanism to kill the virus. But if that becomes very intense, it will affect the brain itself. So the person who can tolerate it and who has the ability of its innate immunity, they can tolerate to some extent and survive. But those who don't have this innate ability, they of course lead to the other end of the life.
एक प्रश्न आहे इथे त्याचा की करोना व्हायरस हा प्राण्यांना का झाला नाही?
See, Ata, yeah, that's a good question and a very hot question because many people ask this, ki why, whether it will affect my pet. Most of the people have the puppies and the cats, kittens. Now, the thing is, one theory which is put that, as I told you, all these viruses are very specific. Now, even a slight change in the ACE receptor may make them accessible to the cell. Like जसं एखादा चोर घरात घुसतोय and he is having a key which is not working under any circumstances. So the lock is such that it will only enter if there is a particular, like cleft in that key. Exactly in the same way, perhaps, of course, this is yet to be studied because the research institute not getting enough funds to study these off-root things because the main focus is on diagnostic and remedies part.
At the
So it is
moment.
predicted that there are receptors present in the animals are slightly different as compared to these humans.
Human body.
That could be the main reason, why the animals are not much affected. But in fact, they are remaining peacefully in the animals. The
Okay.
bats are harboring millions of them, but like their normal flora. Like we also have in our guts many microbes, viruses, but they are our normal flora. Many phages are also there. In the same way, they are present in these other animals, mammals. But once, as I told you in the beginning, whenever these viruses jump from one animal to another animal under circumstances, so they undergo mutation. And if these mutations are harmful, then they kill that or they infect that particular animal host.
So there is another question who asked, how does sunlight kill virus? असं म्हणतात ना थंड प्रदेशामध्ये जास्ती होतो आणि असं म्हणत होते. So there is a question probably in that. How does sunlight kill virus?
See, in general, I can say sunlight have a higher ultraviolet wavelengths, which are of a we can say high energy compared to visible light. So these higher ultraviolet rays have an ability to, you can say, inactivate or demodulate the receptors present on the virus surface. So it is like आपण जसं खेकड्याच्या नांग्या तोडणं म्हणतात ना, खेकडा बाहेर आलं, नांगी तोडून टाकली. In the same way, if these long wavelength of the UV rays in the sun, if they like incident on the say proteins, and certain proteins get denatured on the surface, so obviously this it becomes useless. Means if a key is put and slightly bent little bit because of some action, it will not open the lock. In the same way, on a surface, certain reaction takes place because of the long UV wavelength. it make it useless. Or one more possible mechanism, like in sunlight, if a lot of heat is there, so this heat will denature the protein, because ultimately it is a protein particle. So this denaturation will also kill the microbe, not only microbes but even bacteria.
Mhm. Okay. Uh, is STEM useful during this COVID pandemic? Is STEAM, sorry, STEAM.
Sorry.
Is STEAM useful during this COVID pandemic? Heard the contrary, it may destroy useful symbiotic pathogens that reside in our nasal cavity, thereby disturbing the equilibrium. Can you throw some light? Is a question from Avinash Shirodkar. STEAM.
Yeah, steam is definitely useful if, say, coronavirus is sitting in your upper respiratory tract and steaming is done. So definitely temperature about anything about 45 degrees is supposed to inactivate the virus.
Mhm.
But if there is no virus and if you are daily taking the steam, sitting at home also itself, so it may cause your normal flora also, and it may cause adverse effect on your throat tissue. Because ultimately steam is reaching your tissue also, which is delicate. So that's how.
So it is better not to overdo anything.
If you are coming from outside and doing like then, and if you feel that you are outside for a long time for the essential work, then perhaps you can have a steam. But if you are work from home and just going to shop, and then perhaps it may not be required daily so much. I mean, it should not be like lunch and dinner.
Yes,
uh,
yes. In between,
yeah,
I think last year it had actually
it
become
can
like
be.
that.
Means it should not be practiced, and it may have its own adverse effects. But of course, this is my personal opinion. Means
Yes,
different people have different opinions.
different
Because
opinion.
see, nothing is proved technically. Because even to get virus into the lab and do to BSL-3 facility and create such data is very difficult.
True.
All some bits of data what we have in today is from the original SARS virus
Mm-hmm.
in China, 2012. So we are speculating based on the other RNA viruses.
Okay. So there's another one question from Shreya. Shreya again. Shreya is a microbiology student actually. That's the reason why she's asking so many questions. These questions.
Sure.
As the structure of SARS-CoV-2 is known, can there be a vaccination that would contain a similar non-harmful component that would block the ACE receptor and could act as a preventive measure? The structure of SARS-CoV-2 is known as we know it. Can there be a vaccination that would contain a similar non-harmful component that would block the ACE receptor and could act as a preventive measure?
You mean some chemical to block? Because that blocking the ACE receptor means it is called as a competitive inhibitor usually in our medical science. Like if I have a virus attacking and binding to ACE receptor, at the same time if I put certain chemical, so this is how these different medicines are working. They are RNA polymerase inhibitors. So again, what she's asking, so people are working on it, because since it is a little bit new, there are certain you can say premature data could be available. Because we have, I remember one like in DBT, one the paper has come for assessing, like somebody has put this type of work, but still under research. I can say it's a good way of considering drug mechanism.
Okay, okay. So, of course, a lot of research is still going on, and it may really take some
Yeah,
time to develop
yeah,
that.
it's a new topic actually in that way.
Absolutely. So there is one more question. Can we produce transgenic potato against coronavirus?
Functioning potato against, but what could be the aim? I mean, can that person
Sushant
elaborate a little bit?
Patel, yeah. Sushant Patel is asking this question, and
Yeah,
Sushant, if you can elaborate your question a little bit, maybe we'll be able to answer that. But in the meanwhile, Doctor, now that you have given us so much information about microbiology, microbes, your viruses, and all that. Now tell us in detail about your work in this field I read your bio sketch, but
Okay.
most of the things were not clear to us then. Now I think we'll be in the position to understand it more. Tell us more about the patents that you have worked for, your success stories, and we would like to know
Yeah.
more about you now, doctor, as a
Yeah.
person and
Actually,
as a physician.
yeah, sure. Actually, my work, the projects which are based on, of course, the company. So earlier, I was with a pharmaceutical company called Pyramel. So many of the patents are from that institute, means affiliated to that institute. So there, I was taking care of this drug discovery in anti-infective, anti-cancer, anti-diabetic, and anti-inflammatory from the microbial sources. So we used to get microbes from, you can say, unexplored areas, like you can say hot water springs, cold deserts, and our one of the team member had even visited Antarctica twice, and we collected samples from Antarctica, lichen samples from Antarctica area. He was stationed there for 3 months along with the Indian Antarctic expedition, and we collected samples from there. The whole idea of collecting such, you can say, diverse things from these earth is that we can really get those microbes which have a different metabolism, different way of producing new type of chemicals. And this particular theory that collecting microbes from different area, like the one of the closest unexplored area, or you can say extreme area, is our in Buldhana. There is a crater.
Mhm.
So that crater is, I mean, it is, the history says that it is produced about 5,000 years ago, and it has the water which is alkaline, highly alkaline, up to 12 pH. In the summer, even it goes to 12.5. So when the pH is 12, it means it is alkaline means, I think all of you can understand. It is like our soda. So it is also
Mhm.
called, can be called as soda, you can say. So it has a lot of potassium and sodium salt. Such unexplored areas, they harbor a lot of different microbes. So this particular strategy worked very well. So we collected from deserts like Run of Kutch and such craters and Antarctica, and we collected huge number of microbes. And these microbes we cultivated and checked their metabolism, and because of which we got many, you can say, new drugs.
Mhm.
And these new drugs are under development, like clinical phase 1, clinical phase 2, phase 3. Initially you have to do experiment in the lab, and then in the animals, then you have to go to the patients, like that. So these initial patents are of the various new drugs which we have discovered, and I was one of the key team member.
Oh.
So this is how the patent goes. And later, there are certain drugs which are called as new applications. Like
Mhm.
in this work, we always don't get success immediately. Like when you screen 1,000, we may get perhaps one, and again taking that one ahead is again a probability of getting success is rate is again 1% only. So it's a huge gamble. So in a very intellectual way, we have to screen microbes, we have to screen the compounds. So one example I can give you from Antarctica sample, I had
Mhm.
got a compound which is a powerful anti-TB. Means it is
Anti-TB.
also effective anti-TB,
Mhm.
tuberculosis. Now
Tuberculosis.
it is acting also on the multi-drug resistant TB, which is again a burning issue of the India. Means
Mhm.
perhaps this year India will be number one in TB area also.
Oh.
That's why the anti-TB compounds are really needed, and the therapy itself is very elongated. I mean, long term, one year people have to take the medicines for it.
Mhm.
And I have one compound which is having a very potent activity from the marine sample I had got. So a compound which is having a very potent activity against certain infectious bacteria. And later, when I switched over to a new company called HiMedia,
Mhm.
there we have developed, for especially for Corona, so we have developed a kit based on the antibody by a technique called as ELISA. So for the spike protein, S1 protein, to bind to that S1 protein, there is a specific antibody. So using that antibody and spike protein S1, we have actually developed and commercialized launched a kit called as ELISafe, which is called ELISA kit. Prior to that, I also worked on different types of media for the cultivation of, as I told you, that differential bacteria
Yes.
and selective media.
Media.
So there are certain assays which have developed a super isotonic solution for the microbes, certain changes in the design of the particular media, and we also filed a patent for repurpose of a drug combination for the corona. So that is the latest one which we have, and we are also in the field of hydrocolloid to make India really Atmanirbhar, because in India nobody is cultivating these hydrocolloids.
Mm-hmm.
Everybody is importing, and at a very high cost, and we are having Cadbury's and yogurts at a very high cost. So
Yes.
we put like our company has inculcated, let us make ourselves. So we got certain like develop certain bacteria, and that is how we are working on those. So this is, I can say, in nutshell, I can put this patent history. And of course, on the way there are many publications of the same thing, some stories which could not see the other end. Like we had developed one very powerful anti-diabetic compound.
Okay.
Now that anti-diabetic compound we worked for 3 years.
Okay.
It is from a streptomyces from the Rajasthan region. We had collected the soil and microbes from that area. After working 3 years, we understood that okay, this particular compound is adding obesity to the mice in the female mice. So obviously we have to stop that work. But as a scientific thing, we have published the paper with about 20 authors, so that scientific community will know at least you don't follow this path. Okay, so you can check some other scaffold, etc. So I mean, there are certain many stories and things about this natural product research, the way. And of course, we are good in collaboration. I have a revenue generating collaboration. I have a like perhaps knack, you can say, of writing the projects for the different agencies and contributing to that. So that also, and that's why I get many things for evaluation. So which adds to the knowledge, and knowledge multiplies.
Correct. Correct. Were you always inclined towards microbiology, or it just happened?
No, I mean it. There was an inclination because when in 10th and 12th, I used to read the genetics part, DNA, RNA. So I used to get zapped. Wow, DNA मध्ये एवढं सगळं असतं. That is really fantastic. So I can do this, I can do that, and in happened to that in 12th, you can say May Sutti. So I read a book called Gods from the Outer Space, and this is a story of something different. But in related to that, there is, I don't remember the name of the book, but that is about extrapolating the genetics work, and which was mentioned at what level genes can be taken. So I was wondering, आता genetics करायचं जायचं कुठल्या field मध्ये? So then my cousin who was there in MBLU college, much before, very senior to me. So she said you should go to MBLU college microbiology. So I straight away took admission to microbiology,
Okay.
assuming that I will like to get study about DNA at that time.
So you
So
did
then
your
it started.
microbiology from which college?
MVLU.
From MBLU you did. Okay. Okay.
MVLU म्हणजे चिनाई कॉलेज, अरुण बेडेकर.
Our sister college.
Yes. अच्छा हा, your sister. बरोबर.
With
Yes,
Arun,
yes.
and me and Arun were classmates in
Okay.
Chennai.
Right.
Yes, yes. Doctor,
And
you
then
have
we loved microbiology.
yes, you were saying.
Then, of course, that department was very good, and I really got interested in microbiology, especially medical microbiology and biochemistry.
Mm-hmm.
The path is a little bit changed from molecular biology, but now the application is coming to medical microbiology only. But after PhD,
one thing I missed in this entire thing is how is microbiology and biotechnology? How are they related?
okay.
Are they related, connected, or what is biotechnology?
See, actually, biotechnology is a very compound type of science, which is, say, technically is defined as a science, a facet of biology, which actually gives something productive from the biological system and through the technological advances. But to be fact, it involves microbiology, it involves molecular biology, it involves a lot of bioengineering or engineering mechanical skills. So it's a multifaceted science. But the story of biotechnology, somewhere the core is microbiology, because the techniques which are developed in the microbiology, classical things like with the microbes, those were applied in the animal tissue, plant tissue, and then it becomes a separate entity called biotechnology. Like, a particular level, it is difficult to hold everything under microbiology. It becomes like specialized science. So it come out as a, now today it has multifaceted. Today there are courses called pharmacology, biotechnology, food microbiotechnology, because everything got more and more specialized. But the core is micro. Yeah.
Okay.
Yes.
You have been a mentor to a lot of students, school, colleges. What advice can you give career-wise in the field of microbiology, biotechnology, or biology as a whole?
See, in today's world, like biotechnology is said to be a sort of a revolutionary science in the today's 21st century. So those who really have an inclination from the biology side, so if they can take the career in biotechnology, especially like after 10th, they can act for the science, and there are good number of colleges for biotechnology and microbiology. And but only thing is that whoever want to go for biotechnology, microbiology, they should not stop at BSc level, like graduation level. The real zest of the science will come only when you go for postgraduate or doctorate level, because after BSc again it becomes stagnant, because it is an advancing field, and it is so advancing that sometime now, as I'm on many syllabus committees, now teachers are worried now to include this part is very difficult. So the biotechnology itself is so advancing that every year new things get added, and especially with respect to applications, diagnosis. So that has to be covered, because again the students who are studying, they are evaluated by the companies, so they have to get the new things. That's why it has an advanced science. That's why I say that students who want to take, they have a like knack in biology, they can go to biotechnology, microbiology courses, but keep an eye that they should do minimum post graduation, because the advanced studies will come only after that. Basic science, BSc level, it is not expected.
Correct. I hope Shreya and the others are listening to this, and I'm sure you all are not going to stop, and you all are going to take doctor's advice and really progress on your path. In general, डॉक्टर काय ऍडवाइस द्याल ह्या करोना काळामध्ये आणि even otherwise, related to viruses and all that? What is in general message that you will give to people like us to deal with these viruses?
Yeah. See, first of all, जसं बऱ्याच मी ऐकलं असेल, first of all, like, don't give too much attention to all the news about the coronas, okay? Because these are all, if I pick up from one area, they add some masala to that, you make second news. It is read and add some masala, become the third news. But for a better survival, you should trust your own body, do exercise, do balanced food diet, don't eat exorbitantly, have a balanced and good diet, so that your immunity power remains boosted. Don't get too much tensions, work from home judicially, and of course you have to take vaccine. Don't miss vaccine whenever your turn comes, age wise. Okay, immediately grab the opportunity, and don't you can say a victim to the wrong newses. And if at all you want good information, always follow ICMR site, one of the fantastic website maintained in India today. They have almost every day beautiful updates about everything. So if you want any update, even medicine or even antibiotic, visit the icmr.com. You will see all the good information into that. So stay safe
So just
and enjoy.
last one question, because you said get
Sure.
yourself vaccinated. Again, there is a big controversy about which vaccine to take and which vaccine is good. So असं काही खरंच आहे का की both have their own sort of advantages or properties which can do good to the body?
See, both vaccines clinical data is very good. There is nothing like and even if suppose if you read somewhere, okay, Covaxin is good or Covishield is good, but you have no choice. When you go to the center, you cannot say mujhe Covaxin dena.
Oh.
So trust in the science, because if already more than 5 crore vaccines are given in both categories, so obviously there is something. And India, there are, compared to outside, you can say very less number of things, issues are reported. And of course, side effects are very common, getting fever, fatigue, 1 or 2
Yes.
days. That is not a like bad effect. It is a good effect. It shows that it's working. But other than those two, nothing else. Of course, there are reinfections. There are a good number of reinfections also reported. But these reinfections could be because of the mutant, I said, say B1617. If it has double mutant, I can say. So if it is because of that, of course, yet it is yet to be decided. If the load is so high that it is not taken care by the vaccine antibodies, obviously person get reinfection. And also one important thing I should mention here, that if you take vaccine up to 10 days, you will not get any detectable antibodies, because we have done it ourselves, because we have the kits and we have monitored also. So after taking vaccine, after 1 week, if certain person get corona, and अरे की vaccine चा काय फायदा झाला? But understand the mechanism how it works. Don't blame immediately, unknowingly. Because there is a science. Vaccine taken means you are not made ajramar against corona. 10 days now, antibodies start producing after 10 days. Then it reaches peak up to 28 days. Then it slowly declines. And based on that, they have decided the booster dose. But booster dose, second dose, is very important. That will give us a good memory cell to the body, and it may remain up to 6 months, and even people say 1 year. So that's why it is going to a continuous business till this corona is rooted out from the India, or from the land, you can say, like other viruses in the past. And even good news like Sputnik, Moderna, they are coming out with a fantastic data that people who are infected with corona, for them, if a dose is given, so there is a sudden increase in the antibodies. So for such people, only one dose is good enough. This is a very latest science wire papers which you have got data. Like there is a possibility that those who are infected earlier, there is a possibility that only one dose may be possible.
मला करोना होऊन
Sorry.
गेला.
Yes.
एक डोस
One
झाला,
dose was still good.
याची गरज
Yeah.
नाही.
And they had given a good data of that. There is 1 in 1,000 titer of the antibodies produced after second dose.
Okay.
It's a fantastic data. So there are some silver lines which are being taking place in the remedies and viruses.
In viruses, yes. So all this information also we can get from ICMR site.
Yes. Yeah. ICMR site. ICMR site will tell you about India in general.
Okay.
But if you want some other information, of course, there are science wires or news wires are there. Mediscape is one of the very authentic, which is giving this. And Lancet is one of the journal which is publishing a very authentic data. It's a peer-reviewed journal.
Okay.
But India information, ICMR, you can trust always. Number one. Yeah.
Okay.
Yes.
Thank you so much, Doctor. There would be some questions which are unanswered or sort of unasked by me just now, because there was a lot of scrolling that was happening, and Doctor was also giving a lot of information. So some of them I thought was already answered by Doctor. But if there is any information or any questions which is unanswered, I would request Doctor to quickly afterwards, later on, go through it, and you can always come and see if your question is answered there. And Doctor, I have to really, really thank you for this lovely, wonderful information, the lovely session that we had on behalf of myself and behalf of all our audience. And thanks to the audience also for being, for making this interactive, giving us a chance to ask Doctor questions and, bringing out more information from Doctor. So it was a lovely session, Doctor. Thank you so much.
Thank you so much.
Keep waiting for more such sessions on Expert Speak on my channel. Thank you so much. Have a lovely evening, all of you all.
Okay, thanks to Sonal, and thanks to all viewers. Bye. Bye.
Thank you. Bye bye. Thank you. Thank you.